Namaste everyone. Hello my dear students. Welcome back to online classes brought to you by Vivekananda PU College. I am Supreeth K.C. In today's session, we are going to talk about the nucleic acid under the chapter called as biomolecules. Nucleic acids. Nucleic acids. Dear students, in previous classes we have seen in any living organism you can find out the presence of organic substance as well as inorganic substance. On a biological point of view, we have classified the organic as well as inorganic substance as carbohydrate, proteins, fat, nucleic acid, etc. Okay. In that we have studied how do we analyze the chemical constituent. It may be your organic material as well as inorganic. Even we have seen the complete story of carbohydrates, fruit, proteins, etc. Today we are going to talk about nucleic acid. So what exactly you mean by nucleic acid? Dear students, you already know the nucleic acid. It is a complex organic material, found in a living organism, found in a cell. It is a complex organic material found in the cell. Complex organic material, okay, I'll discuss this word in little bit later. Complex organic material, okay. What exactly you mean by complex organic material? We'll see that later. So the definition of nucleic acid goes like this. It is a complex organic material found in a living cell called as nucleic acid. The examples of nucleic acids are DNA as well as RNA. DNA is known as deoxyribonucleic acid. Deoxyribonucleic acid. RNA is known as ribonucleic acid. Deoxyribonucleic acid as well as ribonucleic acid. These are the examples of nucleic acid. If I say nucleic acid, then it must contain three components mainly. Okay. If I say I have a nucleic acid, then it should have three components mainly. What are the three main components? Okay, we'll see that. The first one, the nucleic acid, okay, it should have a nitrogenous base. Nitrogenous base. There should be a presence of nitrogenous base. Okay, and you should witness the presence of a sugar which is a pentose. Pentose means a sugar with the five carbon atoms. Okay, sugar with the 5 carbon atoms, like this, 5 carbon atoms. Pentose. Okay, and the third one, you should have a phosphate group. If you have these three, the nitrogenous base, sugar which is a pentose, and a phosphate group, then you can say this is a nucleic acid. Dear student, you must know here, when I say it is a nitrogenous base, okay, the nitrogenous base, it is a heterocyclic. The structure is a heterocyclic, like this, heterocyclic. The structure is a heterocyclic. When I say a sugar or a which is a pentose, if I say, then it should be a monomer or monosaccharide, which is a monosaccharide. Sugar is a pentose as well as it is a monosaccharide. And you should have seen the presence of a phosphate group which is contributed by the phosphoric acid. Phosphoric acid. Phosphoric acid. There should be the nitrogenous base is a heterocyclic compound. The sugar, which is a pentose, is a monosaccharide, and a phosphate group is a phosphoric acid. If you have these three, then you can clearly say you will be having a nucleic acid. Nucleic acid. The nucleic acids is classified, or it is having a category as 2. Classified as 2. Okay. What is that? The first one is called as the purines. The second one is called pyrimidines. Purines. Purines. Pyrimidines. Purines and pyrimidines. Purines as well as pyrimidines. These are the two main classes. Purines as well as pyrimidines. Purines as well as pyrimidines. Okay. Purines as well as pyrimidines. Fine. These are the main two classes of nucleic acid. Fine. Sorry, nitrogenous base. Two main classes of nitrogenous base. These are the two main classes of nitrogenous base. Nitrogenous base. Where can I write? Yes, nitrogenous base. Nitrogenous base. The first one, which is a purine. This purine containing the chemicals or the chemical compound called as adenine. Adenine as well as guanine. Adenine as well as guanine. Adenine and guanine are the chemical compound which comes under purine, which is one of the type or one of the category of nitrogenous base. The second one is pyrimidines. Pyrimidines, where you can find out the presence of the first one, cytosine. 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 Even uracil. Cytosine, uracil, as well as thymine. Thymine. Okay. Cytosine, uracil, thymine, they are the examples of pyrimidine. Adenine, guanine is the examples of purines. These are the two main classes of nitrogenous base. In a DNA, in a RNA, you can find out these chemical compounds. Okay. This is about the nucleic acid as well as the nitrogenous base. Okay. In the case of DNA, in the case of your DNA, you can find out the presence of what is this? ATGC. A means adenine. T means thymine. G means guanine. C means cytosine. DNA, you can find out the chemical compound which is a nitrogenous base as this: adenine, thymine, guanine, cytosine. Okay. What about your RNA? In RNA, of course, you can find A means adenine, and of course G means guanine, of course C means cytosine. What about T? T is being replaced by U. U means uracil. This is uracil. In RNA, AUGC you can find out. AUGC. Always, okay. This is the ATGC is a chemicals you can find out in DNA, and even RNA, AUGC chemicals you can find out. Nitrogenous base you can find out. As I told you, dear students, when I say a nucleic acid, there should be a presence of a phosphate group, nitrogenous base, as well as a simple sugar, right? So I have mentioned here adenine, correct? So here this is the structure of adenine. Structure of adenine. This is the structure of adenine. Okay. And I have a pentose here. Pentose. Okay. This is a pentose sugar. In a pentose sugar, the adenine is situated is one of the position at the one of the position of a pentose. Adenine is situated at the one of the position of a pentose. So this structure is called as adenosine. Adenosine. Okay. Adenosine. Now same structure, but one more group is being added, right? One more group is being added, nothing but the phosphate group. This is known to be the adenoic acid. This is known to be the adenoic acid. Okay, wait, I'll write there. Adenoic acid. Adenoic acid. Okay. Even I can find out the presence of a phosphate. So I can see here the only nitrogenous base I can witness, only nitrogenous base, plain nitrogenous base, nothing but the adenine. Here the adenine is situated at the one of the position of a pentose, known to be the adenosine. Here the same structure, but you have one more group been added, nothing but the phosphate group is being added here. So this is called adenic acid or adenylic acid. Adenoic acid or adenylic acid. This is adenylic. Adenylic acid. The nitrogenous base with the sugar is called as nucleoside. Nucleoside. Okay. The nucleoside with the phosphate group called as a nucleotide. I repeat. The nitrogenous base with the simple sugar called as nucleoside. Nucleoside with the phosphate group called as nucleotide. Adenine is one of the example of purines. This is the structure of uracil. Uracil, one of the example of pyrimidines. Okay. If I need to write a nucleoside of uracil, the structure will be the same. Here you can witness the group of uracil, this structure here. Okay, same CS2OH. This is uracil, uracin or uridine, uracin or uridine. Uracin or uridine. If this OH group is being replaced by your phosphate group or phosphoric acid, if the reaction is taking place, then this is known to be the urinoic acid. or uridinoic acid, or uridinic acid. This is about your nucleic acids. This is about your nucleic acid. What are the significance? What is the importance of nucleic acid in our body? What is the importance of nucleic acid in our body? Significance of nucleic acid. We'll see. Significance of nucleic acid. Significance of nucleic acid. So, dear students, you must know nucleic acid is very important for the living organism, because the nucleic acid which act as a genetic material in our body, okay, nucleic acid which act as a genetic material, in our body. The second point, the structure of the cell, the function of a cell, even the whole function, physiological function of the body is regulated by our gene, chromosome, etc. Okay, the primary constituent of this regulatory process is our nucleic acid. Okay, so the function, the structure as well as the functions of the cell regulated by the nucleic acid. Okay, this is about the DNA, deoxyribonucleic acid, which is the genetic material have in our body. The third one, the RNA. Which is a RNA. The RNA in some organisms or in some virus, RNA is a genetic material. In some virus, RNA is a genetic material, okay, which is very important for the survival of a virus, as well as the RNA in our animal body. They are very important, which is called as a mRNA. They are very important for the synthesis of a protein. Protein synthesis. For protein synthesis, the RNA is very, very important. These are the significance of nucleic acid. The genetic material, they act as a genetic material, as well as the structure, as well as the functions of the cell is regulated by the DNA. Even in the case of RNA, which is a genetic material of virus, and in animal cell they are known to be the mRNA, messenger RNA, which is very important for the protein synthesis. This is about the significance of nucleic acid. Okay, let us see the structure of a protein. Okay, the structure of protein. What is the structure of protein? How the structure of protein is there? Okay. Structure of protein. Okay. So as we have studied, we have amino acid which act as a monomer for the formation of a long thread-like structure of a protein, right? So here I have a amino acid called as amino acid 1, amino acid 2, amino acid 3, amino acid 4, amino acid 5, amino acid 6, like the same. It is continuing. A huge chain of amino acids are aggregated, and you can find out the production of a protein. Okay, this is a protein. These are the amino acids. Okay, this is a complete protein. So my protein, if I have a linear chain, means one amino acid is linked by another amino acid, is again linked by another amino acid. Like the same, I have a linear chain, a linear straight chain. Okay, then this structure of a protein is called as a primary structure. Primary structure. Okay, so in broadly sense, my proteins are classified as 4. One is a primary protein structure, secondary protein structure, tertiary protein structure, and quaternary. Okay, secondary, tertiary, quaternary protein structure, primary, secondary, tertiary as well as quaternary. So if I have a linear chain of amino acids which are known to be a protein, then that is called as a primary structure of a protein. Okay, linear chain like structure, this is this kind of a structure. Okay, if this linear chain, okay, if they are arranged as a helix manner, okay, right hand helix, okay, if it you know in the like it is like a twisted ladder, okay, right hand twisted ladder, if it like a right hand twisted ladder, or in the structure of helix, like helix, then here you can find out the amino acid like this. Okay, this structure is called as a secondary protein structure or secondary structure of protein. This is a secondary structure, secondary protein structure. Okay, primary linear, this is like helix, the secondary structure. Okay, if the same structure, if it is arranged like a hollow woolen ball, if it is arranged like a hollow woolen ball, like this, this is amino acid like this. Okay, the bonding stabilization also taking place here. Okay, like this hollow woolen ball, this structure is known to be the tertiary protein structure. This one, tertiary protein structure. If the same structure, okay, the hollow woolen ball like structure, the same structure, if it is arranged like a linear string of a spear, okay, if it is arranged like a linear string of a spear, called as quaternary structure. The structure will be like this. Okay, this can be seen in a polypeptide chain. Say we have discussed the whole structure based on a linear polypeptide chain, only one polypeptide chain. There are some protein which is having 3 to 4 subunits. Say in the case of hemoglobin, we have a 4 subunit, 2 alpha chain or 2 beta chain. Nothing but you can find out the presence of a 4 polypeptide huge chain like this. Okay, this is arranged like a linear string, okay, of a spears. If you imagine in a 3D structure, okay, linear string of spears, this structure is known to be the quaternary structure. This is about the structure of protein: primary structure, secondary structure, tertiary structure, as well as the quaternary structure. Okay. So let us see now the bonding, the types of bond, nature of bond we can see in this biomolecules, maybe in your protein, in your carbohydrate, in your nucleic acid, etc. Okay. So first let us discuss about protein. First one, protein. Protein. As you know, you can find out the linear chain of a protein where the monomers like the amino acids are used to aggregate each other and share a bond called as a peptide bond, and you are obtaining a huge chain, right? You take a example one, say you take the example of amino acid called as serine, the amino acid, and cysteine, another amino acid, serine, cysteine, as well as tyrosine, then glutamic acid, Now let us write the structure of serine. Let us write the structure of serine. CH, CO, NH2, no, NH, and here the R position you will be in R position you can find out the CH2OH, okay. This is formed a bond with the cysteine, okay. Let us write the bonding structure. Later let us see the free structure, okay. This shared a bond with the cysteine where you can find out the presence of NH, CH, CO. Here at R position you can find out the presence of CH2 and SH, okay, sulfur. And again this is paired with or show bonding with the tyrosine where the structure is NH, CH, CO. Here at R position you can find out the presence of CH2 and a ring, and here a OH group, this is tyrosine. This is showing bonding with the your glutamic acid. So the structure is like this. CH2, COOH. Okay. Now you carefully observe. Okay, carefully observe. The free structure. This is the bonding structure. The free structure of the same will be like this, no? CH, COOH, here NH2, and CH2OH, right? Okay. The next group also like this, NH2N, COOH, as well as here H, this group CH2SH, correct? Here CHCOOH, and here NH2, here again CH2, a ring, and above here OH group. Same way here NH2, CH, COOH, here CH2, CH2, COOH, right? Like this structure is a free structure. Now you observe, okay, if one H from here and OH from here is used to react, then there should be an elimination of a water moiety, right? There should be a water molecule elimination. The structure is becomes like this, right? Here again OH from here and H from here structure. OH from here, H from here structure. So this is the same structure I have written here. By the elimination of a water molecule, the bond between two amino acids are formed. This bond is called as a peptide bond. Peptide bond. This bond is called as a peptide bond. This is in the case of a protein. What about your carbohydrates? Carbohydrates. Carbohydrates. Let us take one pentaring, okay, CH2OH, H, OH, 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 correct? Here CH2, OH, H, OH, OH, OH. This is structure, correct? Now, carefully observe. Here I have presence of H, presence of OH, presence of H, presence of H, and the presence of H here, presence of OH, presence of H, H. These are the two carbohydrate or hexon, where you can find 6 carbon. Okay. In the adjacent carbohydrate, which is situated near to near, in this case, the H from one group, OH from one group, means H from one carbohydrate, OH from another carbohydrate, they have to react, and the elimination of a water molecule takes place. Okay. Because of the elimination of water molecule, the structure is like this. Structure is like this. This bond is called as a glycosidic bond. Glycosidic bond or glycosidic linkage. This is the bonding pattern in carbohydrate. This is the bonding pattern in carbohydrate. Like this bonding, because of the bonding shared by each monomer or monosaccharide, a huge polysaccharide is formed. Huge polysaccharide is formed. Okay. So now let us see the structure of nucleic acid, okay, or in DNA. What is the structure? Let us see. Okay. As I told you, in DNA, okay, there are two chains are running parallel to each other. In DNA, there are two chains are running parallel to each other. Here, you can find out this is a base, nitrogenous base. This is nitrogenous base, which is linked to your sugar, a simple sugar. This is your simple sugar. And these two structures are linked by phosphate group. So this is known as each nucleotide, one nucleotide, because you can find out the presence of nitrogenous base, a simple sugar as well as phosphate group. So each one is known to be the, yes, known to be the nucleotide. Yes, these are complementary to each other. The nitrogenous base, if I have a purine A, here I have a thymine. So always the A is paired with the T, C is pairing with the G, or G is pairing with the C. Okay, you never find a mismatch in this. A always used to pair with the T, G always used to pair with the C. Okay, A always used to pair with the T with the C double bond, G always pair with the C with the triple bond. Okay, double bond as well as triple bond. Okay, 3 hydrogen bonds it is. Okay, in this case, you can find out the ester bond. Okay, phosphoester bond. Phosphoester bond means, okay, the phosphate group, okay, they are linked to, so the, here you can find out, the phosphate group, okay, is linked to 5th carbon to 3rd carbon. Okay, the phosphate group is linked to one nucleotide to another nucleotide. So here you can find out the presence of a phosphoester bond on both the side of the helices or both the side of the chain. So this is known to be the phosphodiester bond. This is called as a phosphodiester bond. This is the same diagram. Okay, so phosphate here, for the 5th carbon the phosphate is linked, as well as here for the 3rd carbon of the glucose, for the 3rd carbon of the simple sugar, your phosphate is linked. So this is known to be the phosphoester bond. On the both side, on the both helices, you can find out this presence of this phosphoester bond, known to be the phosphodiester bond. So this is about the nature as well as the structure of the different bonds here, bonds in your biological molecules or biomolecules. So we have studied the carbohydrate, the nature of bond in carbohydrate, nature of bond in protein, as well as nature of bond in our nucleic acid. So this much is enough for you today. So tomorrow we will be talking more about this nucleic acid as well as the different pathways. Thank you so much for your patient listening. I am wind up in the class. Stay at home, stay safe, and take care. Read well and have a nice day. Thank you.